Hi there. Thank you for the invitation to speak today. I'm David Kavanagh. I'm Professor of Complement Therapeutics at Newcastle University, and I'm a kidney doctor.
Hello, my name is Anneliza Andreadi. I'm a PhD student at Newcastle University, and my PhD is funded by the Macular Society.
So, I don't need to tell this audience that age-related macular degeneration is very common. It's a leading cause of irreversible blindness in the developed world, with 3% of adults over 65 and 2/3 over 80 developing disease. It's complex, it's multifactorial, and it results in central vision loss. So that's kind of what you would see. That's the angel of the north. That's what we look out on. And if you've got macular degeneration, that's what you see. Now, there are many different risk factors for age-related macular degeneration. As you might expect, age is one of the main ones. But there are other ones such as smoking, cataract surgery, light exposure, diet. But one of the ones we're going to focus on today is genetics. Now, you might ask, why is a kidney doctor talking to you about the eye? This is an eye of somebody with age-related macular degeneration, and those dots at the back of that eye shouldn't be there. They're called drusen, and in that is complement. Now, this is a kidney biopsy, and that yellow staining shouldn't be there either, and that's complement deposited in the kidney, and that's a disease called C3 glomerulopathy. It results in slowly progressive kidney failure. Now, the underlying cause of both these diseases is very similar, because occasionally patients will develop both. So, what do we know about complement and age-related macular degeneration? And the story starts in 1986 with these two brothers that developed a condition called atypical dense deposit disease, or as we'd later call it, C3G. And on that, you can see that dense deposit shouldn't be there. That black line shouldn't be there, and that's complement depositing. Now, then a few years later, a Japanese group looks at the back of the eye and shows some complement deposited there. Now, we didn't know what that meant at that time, but what we did see was this person here with the same kidney disease, C3G or dense deposit disease. He presented with both that and drusen formation, or early signs of age-related macular degeneration. So this was good evidence that complement was involved in AMD. Now, a lot of time passed and not much focus was on the complement system. And then a new technique came along, a genetic technique, and that was the first study that showed genetic changes in the protein complement factor H might be involved in disease, because this common polymorphism, that's a rare variant of the normal, was more common in age-related macular degeneration. And this really opened up a new window of research. Now, what's complement? So, complement is probably one of the oldest parts of the immune system. It's over 500 million years old. The Tyrannosaurus rex had a complement system. So about 250 million years ago, Tyrannosaurus rex had this part of the immune system. Actually, it goes a bit longer. If you go back to the horseshoe crab, that's 450 million years ago, we can tell you that that too had a complement system. So what's the complement system for? Well, it's one of the first things there to combat bugs. Now, it's not particularly specific. You've all heard of antibodies now, but they're very specific to particular organisms. That's why you get vaccinated. The complement system doesn't need that. It is a non-specific system, and you don't need to have seen a bug before. So when a bug arrives, it activates the complement system. And it's like a positive feedback loop. It's like a Catherine wheel. When you set it off, it just goes and goes and goes. And then what it does is it gives off antibacterial proteins. It coats the bug in some special proteins, and it drills holes in those bugs, and it kills them. Great for killing bugs. However, you all know the term collateral damage, and that's when the complement system is overactivated and kills bugs. So because of that, we've got the activation system, that's the accelerator of the complement system, and we've got the brakes. Now, they involve proteins like factor H and factor I, and what they do is they bind to complement and they downregulate it. So you can think of the complement system as having accelerators and brakes. Now, when complement goes wrong, you get disease. If you don't have enough complement, you will get an infection. If you have too much, you can get lots of different kidney diseases, which is why I'm here, but also the eye disease, which I also research because complement's involved in that too. Too much complement's bad, too little is bad. You just need the right amount. Now, there was a series of other research studies then, because these techniques were becoming more easy to use and cheaper, that linked all the evidence to the alternative pathway of complement. That's just what I've shown you. The accelerators and the brakes of the complement system all seem to be involved in age-related macular degeneration. And over the next few years, various studies showed this. Now, back then, genetic sequencing was very costly. When the first human genome was undertaken, it was $1 billion. Very expensive. Now, for a few £100, I can sequence somebody's entire genome. But that was very useful because what that allowed us to do was sequence all these patients and analyze that. And instead of looking at very common variants and looking at a tiny, tiny percentage of the genome, we can look at everything. And what that did was reveal very rare variants, but they caused a lot of damage. They were very high risk variants. Now, what is a mutation? Well, AMD, as I've said, can be familial and it runs in the DNA, and we have a genetics lab. Now, what is DNA? So DNA is the blueprint for everything. It's like the instruction manual for every protein in the body. Now, there's a cell, and in the cell you've got something called a nucleus, and in the nucleus are chromosomes, and that's where the DNA resides. And that DNA is the blueprint for every protein in the body, and it's made up of just 4 letters, A, T, G, and C. And what we do is we look for spelling mistakes in there. Now, there's a spelling mistake. You can see you get one copy from your mum and one from your dad, and when it's overlaid, it should be identical. But do you see there in yellow those 2 different peaks? Well, that's a genetic spelling mistake. Now, unfortunately, when we read your genetic sequences, there are about 3 billion base pairs, which is about 850,000 pages of text, which is about 28,000 Shakespeare plays. That tells you my love of Shakespeare, I suppose, but there's a lot to read. I'm just going to show you the sequence for one gene, factor H, which we look at. There's factor H gene. Well, not really. That's page 1 of factor H. Then there's another 100 pages, and then the end. Did you spot the spelling mistake in there? Maybe not. So that is a gene. Now, I think of genes as the instruction manual. So here we go. This is the instruction manual for the factor H F1 gene. Now, this has an engine and it's got a steering wheel. Now, if your spelling mistake means that you can't see the engine, the car can't work. If there's no steering wheel, it can't go to the right place. However, sometimes spelling mistakes change things, but that still works. You can see here, the car has an engine and has a steering wheel. It might look different, but it does exactly the same thing. That's what Anneliza is going to tell you about later. Because with factor H and other proteins, there are important bits. This bit's the engine, this bit of the protein here, and this bit is the steering wheel. This tells you where factor H should go, and this functions to put the brakes in the complement system. So how do we know if these changes are harmful? Well, we might look and say, how common are they? If they're rare, it might be more likely it's going to be harmful. Does this variant run in families with AMD? i.e., if everybody in the same family has the same change and has developed disease, it's probably important. Sometimes we can measure the levels, and sometimes we can look at computer predictions of this. And this is some of these proteins I've showed you earlier. So this is C3, And we can model these. This is factor H binding to C3, and that's factor I coming in. So that's the brakes coming on to C3. Now, I can show you some mutations here. They're in red and on factor H in yellow in C3. And those mutations are the ones we see in kidney disease, atypical HUS. These ones are the ones we see in age-related macular degeneration. So sometimes we can look and see where a mutation is and predict what disease it might cause, or indeed, does it cause no disease? Now, how do we do this? Well, this is our lab. This is Edwin and Holly, and they're showing our MP round the lab and showing all the various techniques we use and all the equipment we use to be able to do this, because it's quite difficult. And it's really important that we do this because we need to be able to tell you what your genetic sequence means. And that was one of the reasons the Macular Society funded Anneliza to look at this. And she was going to look at one particular protein, factor I.
So for my project, what I wanted to work out is if these spelling mistakes, what they did to patients, how they affected them, and basically if they are important. So there are three ways to do that. I am working with the factor I protein, as David mentioned. So there are three ways to look at that. Either one is the protein is not being made. Two, the protein is being made, but it's not functioning properly. Or three, the protein is being made and it's functioning normally, which means it does not cause the disease. So there is a graph here showing some of these proteins were not made. They resulted in low levels of this protein, so they do cause the disease. However, there were other proteins that were made. So for these proteins, I did more experiments to see what they do. Think of this experiment as one protein is like a rope, then there is a factor H protein which acts like a helper, holds the rope steady, and then there is the factor I protein which acts like a pair of scissors. So if the pair of scissors is broken, then the rope cannot be cleaved. And if the scissors is working properly, then the protein gets cleaved, the rope gets cut, and then the protein works normally. And what this showed us from my results is that indeed some of these proteins work and some do not. So when the band gets cleaved, it disappears, so the rope gets cut, whereas when the protein is not getting cleaved, then the rope stays intact. So some of them indeed did not work, and they do cause the disease. However, the ones that the protein was made and the levels the protein was made and they did work, they are not responsible for the disease. And what I also did is I developed a new assay, and here I put this protein, the C3b, on the tip, and then I flow across the mutants on that machine you see, and then I look at how these three proteins bind to each other. So there is, you can see from the highest peak that the proteins bind well to each other, but there is also reduced binding on some of them, or even no binding at all, which means that these proteins cause disease. And yes. And we have published. Sorry.
And that was one of the key points, because what we've shown there is that we've taken these spelling mistakes from patients, and some of them result in low levels of the protein, so there's not enough break of the complement system to protect your eye. Some of them, the protein's there, but it doesn't function. And then there are some where there is a spelling mistake, but it makes no difference at all. The protein functions normally. Now, why is that important, and why is this an exciting time for complement research in age-related macular degeneration, and specifically geographic atrophy. Now, I've shown you this diagram before. This is C3b, and this is complement, and it's active. And I've shown you that unchecked complement activation leads to these damaging proteins. They're there to kill bugs. However, if you don't have enough brake, it will cause damage, and it will damage the eye. Pegcetacoplan is a new drug, and what pegcetacoplan does is it stops that overwhelming complement drive, that positive amplification loop, that Catherine wheel. It stops that stone dead, and it stops those damaging proteins having an effect on the eye. Now, I can show you that in a bit more detail. So this is C3. So this is the complement protein I've told you about there. And this is C3b now. And what happens is that pegcetacoplan binds to C3b there. So that little stick diagram is pegcetacoplan. That's where it binds, and that stops the complement system. Now, that's the first one. That was the first ever complement drug that was shown to work in geographic atrophy. There's another one out now, works slightly differently, but still blocking the complement system. And there are lots of different drugs in age-related macular degeneration trials looking at geographic atrophy, targeting the complement system. So I think what we can say now is we're entering a new era for complement therapeutics for dry age-related macular degeneration or geographic atrophy. Now, currently, the agents that are out there, such as pegcetacoplan, have a small effect and slow progression. They're not increasing people's eyesight. So, I mean, they're having a small effect, they're slowing things, but we want to do better. We can see they're having an effect, but we want to do better. So, one of the questions we've got is, are we treating early enough? And will it work for everybody, or only a subsection of patients? Because I've shown you that geographic atrophy and age-related macular degeneration is caused by multiple things, only some of which are complement. And there are other non-complement causes that cause this. So it is possible that if you have a non-complement cause of this, these drugs might not work as well. What this research allows us to do is to understand your genetic risk factors. It allows us to look at your genetic code and to decipher it. And these spelling mistakes we find, we say, well, are they important? And if they are, that might allow us to personalize treatment to you. If you have a genetic defect that we know about, we might be able to target that, and we might be able to target early enough to stop blindness. And I think the other thing this might do is improve the next generation of trials in complement therapeutics. And with that, I'll thank my team at Newcastle University, and I should thank Anneliza, and we're happy to take questions.
Thank you so much for that, David and Anneliza. Absolutely fascinating. So I've been thinking about some questions to ask you. And firstly, I want to ask a question about pegcetacoplan, the drug that you talked about. People may know it by its brand name, Syfovre. Now, it's been licensed in the UK, but it was refused a license in the UK and Europe not very long ago. Could you give us your view on why that drug, why we're seeing different views from different regulators on the same drug for AMD? Why the different approach?
Well, I mean, that's a really great question, and it kind of highlights how regulatory decisions kind of vary across the Atlantic. In the US, as you said, it was approved by the FDA in about February 2023, I think, for treating geographic atrophy. However, in Europe, I think it's been there twice now, maybe.
Yes, there was an appeal.
June and September 2024. And they turned it down. And they turned it down not because they said there was a disagreement about the underlying data. They agreed that there was a slowing of geographic atrophy progression. However, the difference was how they interpreted that, because ultimately, for my vision, I don't want my vision to be lost. And actually, what pegcetacoplan didn't show any functional benefit. There was no translatable, clinically meaningful benefit. It didn't stop vision loss. It had this effect on lesion size. And I think what Europe thought was, well, to have a drug that we're going to license, which, let's remember, came with side effects, you want it to have a clinically meaningful benefit. And these are intravitreal injections, so it's not as if they're completely safe. They're as safe as they can be, but you can get eye inflammation with them. And I think there was a risk of maybe some bleeding or some wet AMD afterwards. So I think there was a difference in the of interpretation in the risk balance across the Atlantic. I think the primary data, they accepted it had this effect on geographic atrophy. The difference was, in Europe, we wanted it to make a clinically meaningful benefit to patients and stop vision loss, which is all what we all want.
Absolutely. It's a hard message for people to hear, though, that people in America can get a treatment and you can't here yet. Absolutely.
Although there are side effects, so it's not.
Yep.
I think I can see both sides of the argument.
I understand. Thank you. So I think one of your slides listed quite a few drugs in different phases of clinical trials that are addressing the complement system. Can you talk a little bit about ones that you maybe think are most likely to become approved eventually? Have you got any favourites?
I wouldn't say I have a favourite. I have to be kind of drug agnostic. However, I think there are some interesting developments. I think I can't keep that slide up to date. Every time I update that slide, there's a new one. I mean, I've talked there about wanting to prevent loss of vision, and I think there was a phase 2 trial by Annexion's ANX007 that actually did have a functional readout. Now, that was an early stage trial. It was very short, very small number of patients, but it was probably the first one that showed a functional benefit. So I'd have to highlight that one as well, that looks good. Now, whether that will translate when we get big patient numbers over a longer period of time, I don't know, but it's certainly good news. And I suppose one of the other ones that's in clinical practice is iptacopan, because most of these drugs are actually injections into the eye. And iptacopan is actually an oral tablet. It's very successful in kidney disease caused by complement. And that would have the advantage of it's just a tablet. Now, of course, there are side effects of that, and you're systemically inhibiting the complement system as opposed to locally. But remember, you don't necessarily know exactly where we need to inhibit the complement system to stop disease. So it might have benefits. But again, it's very early days for that one.
It's worth mentioning there's some other oral treatments not addressing the complement system that are quite close in phase 3 trials, and we're really hopeful for those as well.
Yes, yes.
One for you, Anneliza. Do you envisage all those with AMD having genetic testing ahead of starting treatment so that their treatment can be personalised?
So yes, this is what I've been doing for my PhD. So we've been looking at the genetics so we can see which mutations would be harmful and cause AMD. So the end game would be to treat as early as possible and allow personalised treatment for each patient. So genetics are really important for this. So we know when the drugs come out, which patients can get what, and how we can treat each patient. We can provide a personalized treatment for them.
Thank you. And to go back to you, David, something people might be thinking, should people with AMD be concerned about developing kidney disease? And vice versa? That won't be a connection that people have made before, probably.
Well, no. And I mean, clearly the kidney is the second most important organ in the body after the eye. And I am a nephrologist, and the reason I work in the eye as well as the kidney is this genetic link. Now, what I would say is most people with AMD do not develop kidney disease. AMD is multifactorial. and most of the complement abnormalities we see are very small and don't make that much difference. However, in some of these private mutations that Anneliza has been describing, they actually maybe have far more effect in the complement system. Or there's another condition called dense deposit disease where you get antibodies that activate the complement system. So in the highly impactful mutations or in these autoantibodies, people tend to develop kidney disease first and then may get drusen in the eye. And I'm referred a few people each year that have both eye disease and kidney disease. I would stress, however, that this is very rare. Age-related macular degeneration is very common. Kidney disease, especially complement-mediated kidney disease, is fairly rare. So although there is an association, I want to stress strongly that most patients will not develop kidney disease caused by complement. Now, that said, if you do develop kidney disease and eye disease caused by the complement system, such as dense deposit disease, C3 glomerulopathy, there are very effective treatments in the form of pegcetacoplan, which you've just heard about, and iptacopan, which I've mentioned. So it is something to be aware of. It's very fine print, but we have a treatment for the kidney disease, even if you do.
That's great to hear. Thank you. I'm sure that'll put people's minds at rest. Thank you very much, both of you, for being here today.
Thanks very much.
Thank you.